Hello guys, I hope you're fine today. Today we're going to talk about digestion and complete this lesson. As we last time we finished the digestion in the mouth and in the stomach. Today we're going to talk about the intestinal digestion. Before we start our lesson, I want you to subscribe to my channel and hit the notification bell to receive more videos, and if you have any comments, please write them down. Let's start our lesson now. As you know, this is the diagram of the digestive system. Here is we finished the mouth, the stomach, and then we're going to do the small intestine today. This is the small intestine, and we have very important organs here: the liver, and this is the bile duct. The liver secretes the bile, and this is the bile duct, and the bile goes to stored excess of bile is stored in the gallbladder, and this is the pancreas. Okay, it secretes pancreatic juice. Okay, and the pancreas here it's connected to the small intestine. The intestine consists of duodenum, okay, and ileum, and the intestinal wall contains intestinal enzymes, secretes intestinal enzymes. So here the small intestine consists of duodenum and ileum, and it's about 8 meters long. It coils and loops the coils and loops of small intestine are connected together by mesentery. The juices that help to digest the food in the small intestine are: firstly, we're going to take the bile juice. It is secreted by the liver. It doesn't contain any digestive juices. The bile emulsifies the fat in order to facilitate and accelerate the enzymatic action on the fat that don't dissolve in water. So fat is converted into broken down into small droplets of fat, or we say emulsification. This is called emulsification by the bile juice. Can you see this picture? Again, we have the bile duct and the bile and pancreatic duct. The next juice here is the pancreatic juice. It is secreted by the pancreas. The pancreas contains several things. Number one, it contains sodium bicarbonate, which is alkaline, in order to neutralize the HCl in the stomach and makes the medium alkaline. Pancreatic amylase, it catalyzes the hydrolysis of starch and glycogen into maltose. Okay, this is the first here digestion. Second thing is the trypsinogen enzyme. Trypsinogen enzyme, it's an inactive enzyme. It becomes active trypsin in the duodenum by the enterokinase enzyme that is secreted by the intestinal glands on the lining of the small intestine. Trypsin catalyzes the hydrolysis of protein into polypeptides. So here we have trypsinogen. In the presence of enterokinase enzyme, is converted into trypsin to be activated. So trypsinogen is the inactive form and trypsin is the active form. So trypsin now can work on, it can now break down protein into polypeptides in the presence of alkaline medium. The number 4 here we have lipase enzyme, which catalyzes the hydrolysis of emulsified fat into fatty acids and glycerol. So here again, digestion continues. It has started with the bile. Bile breaks down the fat into fatty droplets, and then it comes here. It continues digestion when where the pancreas secretes this lipase enzyme, and it catalyzes the hydrolysis of emulsified fat into fatty acids and glycerol. Intestinal juices. This is the last thing here. Peptidases. Okay, here polypeptides are broken down into amino acids in the presence of peptidases enzyme in the presence of alkaline medium. Second thing, we have enzymes that convert disaccharides into monosaccharides. So maltose is converted into 2 molecules of glucose in the presence of maltase enzyme. Sucrose is converted into glucose and fructose in the presence of sucrase. Lactose is broken down into glucose and galactose. The last thing here, we have the enterokinase enzyme. Okay, the enterokinase enzyme it's not from the digestive enzymes. They act only as a coenzyme to activate the trypsinogen enzyme. Okay, you have to know this enterokinase, it's very important. I hope that you enjoyed the video, and let's meet again with another video next time. Thank you.